Chapter 2 Biological Classification 2.1 Kingdom Monera, 2.2 Kingdom Protista, 2.3 Kingdom Fungi, and 2.4 Kingdom Plantae, 2.5 Kingdom Animalia, and 2.5 Viruses, Viroids, and Lichens. Chapter 2 Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific, but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdoms was developed, and that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms in plants and animals was easily done and it was easy to understand, but a large number of organisms did not fall into either category. Hence, the two-kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, method of reproduction, and evolution relationships, etc. Classification systems for the living organisms have hence undergone several changes over the time, though plant and animal kingdoms have been a constant under all different systems. The understanding of what groups or organisms be included under these kingdoms have been changing. The number and the nature of other kingdoms have also been understood differently by different scientists over the time. Table 2.1 Classification of 5 kingdoms. Characters 5 kingdoms. Cell type Monera, prokaryotic, protista, eukaryotic, fungi, eukaryotic, plantae, eukaryotic, animalia, eukaryotic. Cell wall Monera, non-cellulosic polysaccharide plus amino acid. Protista present in some, fungi present with chitin, plantae present cellulose, and animalia absent. Nuclear membrane Monera absent and protista present and fungi present, plantae present and animalia present. Body organization cellular, prokaryote, protista cellular, fungi multicellular, loose tissue. Plantae tissue or organ, and animalia tissue or organ system. Mode of nutrition in Monera autotrophic, chemosynthetic and photosynthetic, and heterotrophic, saprophytic or parasitic. In Protista autotrophic, photosynthetic and heterotrophic. In fungi heterotrophic, saprophytic or parasitic, autotrophic, photosynthetic. In plantae and in animalia heterotrophic, holozoic and saprophytic, etc. R.H. Whittaker 1969 proposed a 5-kingdom classification. The kingdoms defined were named by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. Table 2.1 gives a comparative account of different characteristics of the 5 kingdoms. The 3 domain has also been proposed that divides the kingdom Monera into 2 domains, leaving the remaining eukaryotic kingdoms in the 3rd domain, and thereby 6 kingdom classification. You will learn about this system in detail at higher classes. Let us look at this 5 kingdom classification to understand the issues and consideration that influence the classification system. Earlier classification system include bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and the angiosperms under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which is widely different in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae cyanobacteria with the other groups which were eukaryotic. It is also grouped together the unicellular organisms and the multicellular ones. Say for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between heterotrophic group fungi and autotrophic green plants, though they also showed characteristic difference in their wall composition. The fungi had chitin in their walls, while the green plants had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas, chlorella, either placed in algae within plants and both having cell walls, with paramecium and amoeba were earlier placed in the animal kingdom which lacks cell wall. It has put together organisms which in earlier classification were placed in different kingdoms. This happened because the criteria for classification changed, and these kind changes will take place in future too, depending on the improvement in our understanding of the characteristics and the evolutionary relationship. Over time, an attempt has made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities, but is also phylogenetic, that is based on the evolutionary relationships. In this chapter, we will study characteristics of kingdom Monera, Protista, and fungi of the Whittaker system of classification. The kingdom Plantae and Animalia, commonly referred to as plant and animal kingdom respectively, will be dealt separately in the chapter 3 and 4. 2.1 Kingdom Monera. Bacteria are the sole members of the kingdom Monera. They are also most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot springs, deserts, snow, and deep oceans where very few other life forms can survive. Many of them live on or on the organisms as parasites. Bacteria are grouped under four categories based on their shape: the spherical coccus, plural cocci, and the rod-shaped bacillus, plural bacilli, the comma-shaped vibrium, plural vibrio, and the spiral spirillum, plural spirilla. In figure 2.1, we can see the cocci and bacilli as spores, and the spirilla have flagellum and vibrio. Next, though the bacterial structure is very simple, they were very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is, they depend on the other organisms or on dead organic matter for food. Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty areas, halophiles, hot springs, thermoacidophiles, and marshy areas, methanogens. Archaebacteria differ from other bacteria in having different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cow and buffaloes. They are responsible for the production of methane biogas from the dung of these animals. 2.1.2 Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall, and if motile, a flagellum. The cyanobacteria, also referred to as blue-green algae, have chlorophyll similar to green plants and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial, or filamentous, freshwater or marine or terrestrial algae. The colonies are generally surrounded by a gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in a specialized cell called heterocyst, example nostoc and anabina. Chemosynthetic autotrophic bacteria oxidize the various inorganic substances such as nitrates, and ammonia and use the release energy for the ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume roots, etc. Some are pathogens causing damage to human beings, crops, farms, animals, and pets. And chlorella, typhoid, tetanus, citrus canker are well known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Figure 2.3 shows that, and sometimes under unfavorable conditions they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. 2.2 Kingdom Protista. All single-cell eukaryotes are placed under protista, but the boundaries of the kingdom are not well defined. What may be a photosynthetic protistan to one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans under protista. Members of protista are primarily aquatic. This kingdom forms a link between with the other dealing with plants and animals and fungi. Being eukaryotes, the protistan cell body contains a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protista reproduce asexually and sexually by a process involving cell fusion and zygote formation. In 2.2.1, chrysophytes. This group includes diatoms and loaded algae desmids. They are found in freshwater as well as in marine environments. They are microscopic and float passively in water currents, plankton. Most of them photosynthetic. In diatoms, the cell wall form two thin overlapping shells which fit together as in a soap box. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have well left behind large amount of cell wall deposits in their habitat. This accumulation over billions of earth is referred to as diatomaceous earth. Being gritty, this soil is used for polishing, filtration, and of oils and syrups. Diatoms are the chief producer in oceans. 2.2.2 Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red depending on the main pigments present in their cells. The cell wall has if cellulosic plates on the outer surface. Most of them have two flagella. One lies longitudinally and other transversely in a furrow between the wall plates. Very often dead dinoflagellates, example goniolates, under such rapid multiplication that they make the sea appear red tides. Toxins released by such large numbers may even kill other marine animals such as fishes. 2.2.3 Euglenoids. Majority of them are freshwater organisms found in stagnant water instead of a cell wall. They have a protein-rich layer called pellicle which makes their body flexible. They have two flagella, a short and a long one. Though they are photosynthetic in the presence of the sunlight, when deprived of sunlight, they behave like heterotrophs by predating on other smaller animals. Interestingly, the pigments of euglenoids are identical to those present in higher plants. Example, euglena. 2.2.4 Slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and leaves, engulfing organic material. Under suitable conditions, they form an aggregation called plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiate and form fruiting bodies bearing spores at their tips. The spores possess two walls. They are extremely resistant and survival for many years. Even under adverse conditions, the spores are dispersed by air currents. 2.2.5 Protozoans. All protozoans are heterotrophs and live as predators or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoans. These organisms live in freshwater, seawater, or moist soil. They move and capture their prey by putting out pseudopodia or false feet as in amoeba. Marine forms have silica shells on the surface. Some of them, such as entamoeba, are parasites. Flagellated protozoans. The members of this group are either free-living or parasitic. They have flagella. The parasitic forms causes diseases such as sleeping sickness, example trypanosoma. Ciliated protozoans, they are aquatic, actively moving organisms. Because of the presence of thousands of cilia, they have a cavity gullet that opens to the outside of the cell surface. The coordinated movement of rows of cilia causes the water laden with food to be steered into the gullet. Example paramecium. Sporozoans. This includes diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is the Plasmodium malarial parasites, which causes malaria, a disease which has a staggering effect on human population. 2.3 Kingdom fungi. The kingdom fungi constitute a unique kingdom of heterotrophic organisms. They show a greater diversity in morphology and habitat. You must have seen fungi on a moist bread and rotten fruits. The common mushroom you eat and toast will also fungi, while spots seen on mustard leaves are due to parasitic fungus. Some unicellular fungi, example yeast, are used to make bread and beer. Other fungi causes diseases in plants and animals. Wheat rust causing Puccinia is an important example. Some are the sources of antibiotics, example penicillin. Fungi are cosmopolitan and occur in air, water, soil, and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infections. With the exception of yeast, which are unicellular fungi or filamentous, their bodies consist of long, standard thread-like structure called hyphae. The network of hyphae is known as a mycelium. Some hyphae are continuous tube filled with multinucleated cytoplasm. These are called cenocytic hyphae. Others have septate or cross walls in their hyphae. Their cell walls of fungi are composed of chitin and polysaccharides. Most of the fungi are heterotrophic and absorb soluble organic matter from dead substrates, and hence are called saprophytes. Those that depend on living plants and animals are called parasites. They can live as symbionts in ocean with algae as lichens and with roots of fire plants as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction is by spore called conidia or sporangiospores or zoospores, and sexual reproduction is by oospores, ascospores, and basidiospores. The various spores are produced in a distinct structure called fruiting bodies. The sexual cycle involves the following three steps: 1. Fusion of protoplasm between two motile or non-motile gametes called plasmogamy. Fusion of two nuclei called karyogamy. Meiosis in zygote result in haploid spores. When a fungus reproduces sexually, two haploid hyphae or compatible mating types come together and fuse. In some fungi, the fusion of two haploid cells immediately result in diploid cells 2N. However, in other fungi, Ascomycetes and Basidiomycetes, an intervening dikaryotic stage N plus 1 2 nuclei per cell occurs, and such condition is called dikaryon, and the phase is called dikaryophase of fungus. Later the parental nuclei fuse and the cell become diploid. The fungi form fruiting bodies in which reduction division occurs, leading to formation of haploid spores. The morphology of mycelium, mode of spore formation, and fruiting bodies form the basis for the division of the kingdom into various classes. 2.3.1 Phycomycetes. The members of Phycomycetes are found in aquatic habitats and on decaying wood in moist and damp places, or as obligate parasites on plants. The mycelium is aseptate and cenocytic. Asexual reproduction takes place by zoospores, motile or by aplanospores, non-motile. These spores are endogenously produced in the sporangium. A zygospore is formed by fusion of two gametes. These gametes are similar in morphology. Isogametes are dissimilar. Anisogametes are oogametes. Some examples are mucor and rhizopus, bread mold mentioned earlier, and albugo, the parasitic fungi on mustard, and Ascomycetes, commonly known as sac fungi. The Ascomycetes are mostly multicellular. Example Penicillium are rarely unicellular. Yeast and Saccharomyces, and they are saprophytic decomposers, parasitic or coprophilous, growing on dung. Mycelium is branched and septate. The asexual spores are conidia produced exogenously on the special mycelium called conidiophores. Conidia on the germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in sac-like asci, singular ascus. These asci are arranged in different types of fruiting bodies called ascoscops. Some examples are aspergillus and claviceps and neurospora. Neurospora is used extensively in biochemical and genetical work. Many members like morels and truffles are edible and are considered delicacies. And Basidiomycetes 2.3.3, commonly known as Basidio, forms of Basidiomycetes are mushrooms, fungi or puffballs. They grow in soil, on logs and tree stems, and in living plant bodies as parasites. Example rust and smuts. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought by about fusion of two vegetative or somatic cells of different strains or genotypes. The resultant structure is dikaryotic, which ultimately gives rise to basidium. Karyogamy and meiosis takes place in the basidium, producing 4 basidiospores. The basidiospores are exogenously produced on the basidium, plural basidia. The basidia are arranged in fruiting bodies called basidiocarps. Some common members are agaricus mushroom and Estilagos mud and Puccinia rust fungus. 2.3.4 Deuteromycetes, commonly known as imperfecti fungi. because only the asexual or vegetative phases of these fungi are known. When the sexual forms of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stage have been given on one name that and placed under Deuteromycetes, and the sexual stages another and placed under the another class. Later, when the linkages were established, the fungi were correctly identified and moved out of the Deuteromycetes. Perfect sexual stages of the members of Deuteromycetes were discovered. They were often moved to Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce only by asexual spores known as Conidia. A mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposers of litter and help in mineral cycling. Some examples are Alternaria, Cholicotrichum, and Trichoderma. 2.4 Kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll containing organisms commonly called plants. A few members are partially heterotrophic, such as the insectivorous plants or parasites. Bladderwort and Venus flytrap are examples of insectivorous plants, and Cuscuta is total parasite. The plant cell have an eukaryotic structure with prominent chloroplast and cell wall mainly made up of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plantae include algae, bryophytes, pteridophytes, gymnosperm, and angiosperms. Life cycle of plants has two distinct phases: the diploid sporophyte and the haploid gametophytic, that alternate with each other. The lengths of the haploid and diploid phases and whether these phases are free living or dependent on others vary among different groups in plants. This phenomenon is called alternation of generation. You will study further details of this kingdom in chapter 3. Kingdom Animalia 2.5. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cells lack cell walls. They directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food reserves as glycogen or fat. Their mode of nutrition is holozoic by ingestion of food. They follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Certain phases of various phyla are described in chapter 4. Viruses, viroids, and prions and lichens. In the five kingdom classification of Whitaker, there is no mention of lichens and some acellular organisms like viruses, viroids, and prions. These are briefly introduced here. All of us who have suffered the ill effects of common cold or flu know what effect viruses can have on us. Even if we do not associate it with our own condition, viruses do not find a place in classification since they are not considered truly living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having a inert crystalline structure outside the living cell. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Would you call viruses living or non-living? Viruses means venom or poisonous fluid. Dmitry Ivanovsky 1892 recognized certain microbes as casual organisms of the mosaic disease of the tobacco. These were found to be smaller than bacteria because they passed through the bacterial-proof filters. M.W. Bezernik 1898 demonstrated the extract of the infected plants of tobacco could cause infections in healthy plants and named the new pathogen virus and called the fluid as Contagium vivum fluidum, infectious living fluid. W.M. Stanley 1935 showed that viruses could be crystallized and crystals consisted largely of proteins. They are inert outside their specific host cell. Viruses are obligate parasites. In addition to proteins, viruses also contain genetic material that could be either RNA or DNA. No viruses contains both RNA and DNA. A virus is a nuclear protein and the genetic material is infectious. In general, viruses that infect plants have single-stranded RNA and viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. Bacterial viruses are bacteriophages. Viruses that infect the bacteria are usually double-stranded DNA viruses. The protein coat called capsid, made of small subunit called capsomeres, protects the nucleic acid. These capsomeres are arranged in helical or polyhedral geometry forms. Viruses cause diseases like mumps, smallpox, herpes, and influenza. AIDS in humans is also caused by virus. In plants, the symptoms can be mosaic formation, leaf rolling, uncurling, yellowing, and veiny clearing, dwarfing, and stunted growth. Viroids. In 1971, Theo Dainer discovered a new infectious agent that was smaller than virus and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein coat that is found in viruses, hence the name viroid. The RNA of the viroid was low of molecular weight. Prions in modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. The agent was similar in size to viruses. These agents are called prions. The most notable diseases caused by prions are bovine spongiform encephalopathy, commonly called mad cow disease in cattle, and it is analog variant Rutherford-Jacob disease in humans. Lichens are symbiotic association, that is mutually useful association between algae and fungi. The algal component is known as picobiont and the fungal component is as mycobiont, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi and fungi provide shelter and absorb mineral nutrients and water for its partner. So close is the association, that is one saw a lichen in nature on wood. Never imagine that they have two different organisms within them. Lichens are very good pollution indicators. They do not grow in polluted areas. Summary. Biological classification of plants and animals were first proposed by Aristotle on the basis of simple morphological characters. Linnaeus later classified all living organisms into two kingdoms, Plantae and Animalia. Whittaker proposed an elaborate five-kingdom classification, Monera, Protista, Fungi, Plantae, and Animalia. The main criteria of the five-kingdom classification were cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. In the five-kingdom classification, bacteria are included in kingdom Monera. Bacteria are cosmopolitan in distribution. These organisms show the most extensive metabolic diversity. Bacteria may be autotrophic or heterotrophic in the mode of nutrition. Kingdom Protista includes all single-celled eukaryotes such as Chrysoprasids, Physalis, Euglena, Stymos, and protozoans. Protista have a definite nucleus and other membrane-bound organelles. They reproduce both sexually and asexually. Members of kingdom fungi show greatest diversity in structure and habitat. Most fungi are saprophytic in their mode of nutrition. They show asexual and sexual reproduction. Mycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes are the four classes under this kingdom. The Plantae includes all eukaryotic chlorophyll-containing organisms. Algae, bryophytes, pteridophytes, gymnosperm, and angiosperms are included in this group. The life cycle of plants exhibit alternation of generation, gametophytic and sporophytic generations. The heterotrophic eukaryotic multicellular organisms lacking a cell wall are included in this kingdom Animalia. The mode of nutrition of these organisms are holozoic. They reproduce mostly by the sexual mode. Some acellular organisms like viruses and viroids as well as the lichens are not included in the five kingdom system of classification.